DNA is great and all, but how the heck does it get from, that spindly, helixy thing to an actual human body? We know that DNA codes for the formation of proteins, however, it kind of has trouble making proteins because it's stuck in the nucleus. So to compensate for this, DNA goes under a process called transcription whenever it wants to produce a protein. It's a three-step process, and what happens first is, during the first step, the initialization phase, RNA polymerase comes along until it scans the DNA until it finds a thing called a Tata box. A Tata box is basically the genetic version of a green light. It basically means go, start here, go now. And so RNA polymerase takes the Tata box and it attaches itself to the DNA at that spot, and it starts traveling along the DNA from a 3' to 5' direction. And it gathers complementary pairs and starts creating another RNA strand. Eventually, the RNA polymerase will hit a thing called a termination codon. We'll specify what that sort of is a little bit later. And so by the end of it, it separates, and you have this long amino acid chain. It usually starts with an AUG, or a methionine codon, and then it ends with a terminator codon, which is usually a UAG, or a UGA, or a UAA. And from here, actually the RNA isn't done yet. There's still a couple things that needs to be fixed. So the spliceosome comes along. I love that word, spliceosome. And first of all, it attaches a 5' cap to one end of the RNA. That protects that side of the strand. And it attaches a poly A tail to the other. It's basically just a long sequence of, guess what, A's. And this is so the DNA is protected against, hungry enzymes that might be traveling by. And finally, what happens is the spliceosome cuts out any introns that are in the RNA code. Introns are basically just pieces of genetic code that don't actually code for anything. They're just there. And so by the end of this process, you have an mRNA strand, which is compiled, it's shortened, it's protected, and it's just basically a piece of RNA that's ready to go out and undergo the next phase of the gene expression. And this phase is called hold on, sorry, it'll hit me, translation. And so basically at this point, the mRNA, it travels outside of the nucleus and ends up in the cytoplasm, and it meets a ribosome. A ribosome, we've learned about this before. It's floating around in the RFER and all that. And a ribosome is just basically a protein built in with some rRNA, or called ribosomal RNA. And so the mRNA attaches itself to the ribosome, and the ribosome starts on the methionine, the starter codon. And what happens here is, inside our ribosome, ribosome, if I could English today, that would be wonderful. There are these things called binding sites, and here the mRNA is held still while tRNA comes along. It fuses itself to the mRNA. A tRNA is called the transfer RNA, and it's basically on one side it has 3 nucleotides, and on the other end it carries a very specific amino acid, and we'll talk about those a little bit later. And from here on out, this process repeats. The ribosome travels down the RNA strand, and the tRNAs keep coming, keep reattaching, keep detaching, and eventually all of these amino acids are gathered together in a long chain. And by the end of it, you have an amino acid chain, a fully functional protein. Well, I shouldn't say that, because it's still just basically a string. And so this chain enters chaperone. That's a fun name as well. And here, it begins to fold up with those hydrophilic bonds and those other phases that we've already talked about. And all of a sudden, you have a protein that is ready to be used by the body. I may have forgot to talk about this earlier, but that's okay. Um, so a codon is basically just a string of three nucleotides lined up together in order. And you'll see here that each three codons accounts for a type of amino acid from the tRNA strand. And you notice that they can sometimes be a bit redundant. That means that one codon may may produce the same amino acids as another codon. And this protects the amino acid chain. For example, if maybe there's a point mutation in the RNA, or maybe one of the strands is damaged, then there is less of a chance of the wrong amino acid being used, and thus there's a greater chance for the correct protein to be used. It's protection.